In Chapter 14, we begin our discussion of the kidneys. So we begin with a short discussion of renal function. We divide the function of the kidneys up into five different points. Number one is regulation. The kidneys are responsible for regulating the concentration of water found in the body, simply by determining how much water is released from the body, the ion composition of the blood, the pH of the blood, and the overall volume of the blood. And we'll talk during the course of our chapter about how the kidneys accomplish each of these functions. Number two, the kidneys are responsible for removing metabolic waste products from the blood. These include things like urea, uric acid, products of hemoglobin breakdown, and other normal products of metabolism. Number three, the kidneys are responsible for removing some, but not all, xenobiotics from the body. And so these are compounds that wouldn't normally be found in the body, and so would include things like food additives, drugs, pesticides, such as those that might be found on the blueberries. On this particular slide, many of these compounds are removed, not by the liver, but by the kidneys. Number four, we see that gluconeogenesis is also a function of the kidneys under certain circumstances. And so, in certain circumstances, the kidneys are responsible for synthesizing glucose from amino acids and thereby contributing to the maintenance of glucose levels in the bloodstream. And number five, the kidneys are responsible for the production of some hormones and enzymes. And so we've talked about erythropoietin and vitamin D. We'll mention their synthesis again in the course of this chapter. Renin is also secreted by the kidneys, and we'll talk about its function as well during the course of our chapter. In section 14.2, we talk about the major structures that are associated with the renal system. We begin by mentioning that the kidneys are retroperitoneal, meaning that although the kidneys are found in the abdominal cavity, they are actually not within the abdominal cavity, but they are located behind the peritoneum that surrounds the abdominal organs. As we look at our screen, we see that we have a tube extending from the renal pelvis, referred to as the ureter, on both the left and right sides. The ureters make their way down to the bladder and, of course, transfer the recently formed urine to the bladder, where it is stored until it leaves the body via the urethra. We now turn our attention to the actual structures found within the kidney. And as we look at the kidney, what we see is this. So the large central opening inside the kidney, into which the recently formed urine drains, is the renal pelvis. And so, of course, from the renal pelvis, the forming urine makes its way into the ureter. If we make our way backwards from the renal pelvis, we notice these calyces that occur. So right here, right here, right here, right here are renal calyces. And so these are the openings into which the collecting ducts empty. We'll, of course, be talking about the collecting ducts shortly. As we look at the remainder of the kidney, what we notice is that although it's not labeled on our diagram, it is labeled on our diagram here. So the outer portion of the kidney is known as the renal cortex. And so what I'm pointing to here, all throughout the outer realm of the kidney, is known as the renal cortex. The inner layer is known as the renal medulla. And so the region that we see here is known as the renal medulla. In a nutshell, what we notice is that each kidney is composed of over 1 million nephrons. The nephrons are the filtering units of the kidney, and the nephrons are arranged mostly in the renal cortex, but some of them dip down into the renal medulla, and these are the units that actually perform the filtering of the blood. The last structure that we neglected to mention here is the renal papilla, and so the renal papilla is found here. It's the structure that's located between the renal medulla and the renal calyx that we mentioned previously. We now look at the structures of a nephron, or at least we look at them in brief on this particular slide. And so we have approximately 1 million of these nephrons per kidney, and the nephrons consist of basically two components. The first component is the renal corpuscle. And we can see the renal corpuscle fairly nicely at this particular point on this diagram. And so the renal corpuscle consists of two components. One is the glomerulus, And so the glomerulus is this collection of capillaries through which the filtration occurs. The second is a structure referred to as Bowman's capsule. And so once again, you can use that fist into a balloon analogy that we used when we talked about the lungs. Imagine taking a fist and pushing it into a loosely inflated balloon. The balloon would fold around the fist, but it wouldn't collapse completely. And so in this particular case, the glomerulus is like the fist and the Bowman's capsule is like the balloon. You end up with this very thin space that's located all around the glomerulus, referred to as Bowman's space, and this is the space into which the so-called ultrafiltrate, which is going to eventually become the urine, filters. So to summarize, the nephrons are the functional units of the kidney. They're the filtering units of the kidney, and we have approximately 1 million nephrons per kidney. If we look in more detail at the structure of a nephron, then we see that the glomerulus is, as I mentioned, composed of these glomerular capillaries. And so we see the glomerular capillaries at the upper right-hand side of our screen, where I'm pointing to at this particular time. And then, as we mentioned, we have this Bowman's capsule that surrounds the glomerular capillaries, and that's what I'm pointing to here. The Bowman's capsule, as we alluded to, has 2 layers. One layer is the parietal layer, and so that's equivalent to the outside of that balloon. The second layer is referred to as the visceral layer. The visceral layer is attached to the surface of the capillaries themselves. The portion of the visceral layer of Bowman's capsule that's attached to those capillaries is specialized. And the cells that are attached to the glomerular capillaries form these structures referred to as podocytes. If we look at the middle portion of our diagram here, then we see these podocytes. And so the cell processes of the podocytes extend, and they almost resemble interlocking fingers. The spaces between the fingers are known as filtration slits. So let's go down here to the very bottom of our diagram and look at what's happening here. And so if you follow the pointer, this is the capillary lumen. Here we have openings in the capillary through which filtrate moves. The filtrate can then move easily through this basement membrane to which the visceral layer of Bowman's capsule is attached. And then it moves through these filtration slits that are in between the so-called foot processes of those podocytes, which are the specialized cells that make up the visceral layer of Bowman's capsule. And so that's the journey that the so-called ultrafiltrate takes. The ultrafiltrate is going to be modified considerably, but eventually what remains after that modification is urine. We also have an afferent and efferent arteriole. The afferent, of course, as I'm pointing to here, delivering blood to the glomerulus, the efferent carrying blood away, and then Bowman's space is what you're left with if you look at the space that's located between the visceral and parietal layer of the Bowman's capsule. We now turn our attention towards the anatomy found in the remainder of the nephron. So let's take a look at the diagram that we have on the screen. And we begin with the glomerulus here, which we've already discussed. We want to trace the flow of this ultrafiltrate that's leaving the glomerular capillaries. The ultrafiltrate leaves and it makes its way into this portion of the nephron known as the proximal convoluted tubule. And so it makes its way through the proximal convoluted tubule, and then it makes its way into this structure referred to as the loop of Henle. Here we have the descending limb of the loop of Henle. Here we have the ascending limb of the loop of Henle. There are certainly there are certainly very specific physiological actions that take place in the descending and ascending limb of the loop of Henle that we'll talk about shortly. We then make our way to the distal convoluted tubule, and finally the distal convoluted tubule terminates in the collecting duct. The collecting duct receives what is going to be urine from a number of different nephrons, as you can see. And then eventually that collecting duct is going to empty into a calyce. As we mentioned, that urine is going to go into the renal pelvis, into the ureter, into the bladder, into the urethra, and then out of the body. It's worth noting as we close this particular slide that there are two regions to the collecting duct. And so the first portion of the collecting duct, the portion towards the outer side of the kidney, is known as the cortical collecting duct. And then as we make our way down closer to the center of the kidney, the collecting duct makes its way into the medullary region of the kidney, and so it's referred to as the medullary collecting duct. Next, we want to be clear that there are different types of capillaries that are associated with the nephron. Not all these capillaries are part of the nephron, but they're simply in the vicinity of the nephron. So the first type of capillary we want to mention are the glomerular capillaries. And so the glomerular capillaries, of course, make up the glomerulus that we've talked about previously, and these may be considered part of the nephron. They're nestled in that Bowman's capsule, as we mentioned, surrounded by podocytes, and they're the process through which filtration actually takes place. The other capillaries that are associated but not part of the nephron are referred to as peritubular capillaries, and so the peritubular capillaries surround the nephron. So it is worth noting that we do go from an afferent arteriole to capillaries to an efferent arteriole to another set of capillaries, and only then do we go back to the heart. The purpose of the function, let's say, of the peritubular capillaries is to reabsorb fluid that's lost during this process of ultrafiltration. As we'll see momentarily here, most of the fluid that is filtered is reabsorbed and not lost from the body in the form of urine. This is necessary because there's a tremendous volume, 180 liters per day, in fact, that is filtered. Interestingly, we have two types of nephrons that are found in the kidney. The majority of nephrons found in the kidney are cortical nephrons. These nephrons are located primarily in the renal cortex, only making their way ever so slightly into the renal medulla. Some of the cortical nephrons don't make their way into the renal medulla at all. You'll notice that it's the loop of Henle that makes its way into the renal medulla in the case of cortical nephrons, if anything makes its way into the renal medulla. The other type of nephron that we have in the kidney that are much less common, only 15% of nephrons or so, are juxtamedullary nephrons. These are nephrons that have loops of Henle that dip deeply into the renal medulla. These are the nephrons that are primarily responsible for concentrating urine. As you look across the animal world, as we've talked about in various biology classes, you see varying degrees of organisms' ability to concentrate urine. Very little in amphibians, but as the organisms become more and more independent of the water, they gain this ability to concentrate urine because they need to not have to have access to water and be drinking water continually. And so this is what's responsible for that concentration, this loop of Henle. And as you look across the animal world, you can see that animals like kangaroo rats, which are world champions at concentrating their urine, emitting what are almost little wet crystals when they urinate, have very extensive loops of Henle. So, two types of nephrons, 85% cortical, 15% juxtamedullary. It's the juxtamedullary nephrons that are responsible for the concentration of urine. The last structure that we'll look at as we talk about nephron anatomy is the so-called juxtaglomerular apparatus, which is shown here in our diagram. The juxtaglomerular apparatus consists of two portions. The macula densa, which is a collection of cells on the upper wall of the distal convoluted tubule. These cells are sensitive to sodium and can detect the amount of sodium that's moving past them. If they detect a decline in the amount of sodium that's present, they secrete paracrine factors that cause the so-called juxtaglomerular cells, which are labeled as granular cells in this diagram that I'm using from the anatomy book since it was better. But these are the juxtaglomerular, also known as granular cells, and they release renin into the afferent arteriole in a mechanism that we'll talk about in more detail a little later in our class. This release of renin eventually leads to the retention of sodium and an increase in blood pressure in the body.